Glycolysis is, a fundamental metabolic pathway. It is the sequence of reactions that converts one molecule of glucose into two molecules of pyruvate. This process releases energy in the form of ATP and NADH. Glycolysis takes place in the cytoplasm of the cell. It does not require oxygen, making it an anaerobic process. This pathway is nearly universal, found in almost all living organisms. It serves as the primary source of metabolic energy for many cell types. The entire pathway can be divided into two main phases. The first is the preparatory phase, which requires an investment of energy. The second is the payoff phase, which generates a net gain of energy for the cell. The preparatory phase begins with the phosphorylation of glucose. The enzyme hexokinase catalyzes this first step. It transfers a phosphate group from an ATP molecule to glucose. This reaction forms glucose 6-phosphate. The phosphorylation traps glucose inside the cell, as the cell membrane is impermeable to the charged molecule. This initial step is irreversible under cellular conditions. It commits the glucose molecule to further metabolism within the cell. This is a critical point of regulation, ensuring that glucose is captured efficiently when it enters the cytoplasm. The energy from one ATP molecule is consumed here to prime the glucose for subsequent reactions. Next, glucose 6-phosphate is converted into its isomer, fructose 6-phosphate. This isomerization reaction is catalyzed by the enzyme phosphoglucose isomerase. This step is a readily reversible reaction. It involves changing the 6-membered glucose ring into a 5-membered fructose ring. This structural rearrangement is necessary for the next phosphorylation step to occur symmetrically. The enzyme specifically recognizes the substrate and facilitates the shift in the carbonyl group from carbon 1 to carbon 2. This sets up the molecule for cleavage into two 3-carbon fragments later in the pathway, which is a key strategic goal of this initial phase of glycolysis. The third step is another phosphorylation event. The enzyme phosphofructokinase 1, or PFK1, adds a second phosphate group. It uses another molecule of ATP to convert fructose 6-phosphate into fructose 1,6-bisphosphate. This reaction is the major commitment step of glycolysis. It is essentially irreversible under physiological conditions. PFK1 is a key regulatory enzyme in the pathway. Its activity is tightly controlled by various allosteric effectors, which we will discuss later. This second energy investment further destabilizes the sugar molecule, preparing it for the cleavage that follows, marking the end of the initial energy investment stage. The preparatory phase concludes as the 6-carbon fructose 1,6-bisphosphate is cleaved. The enzyme aldolases splits this molecule into two different 3-carbon molecules. These products are dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, G3P. This cleavage reaction is reversible. It represents the point where one 6-carbon pathway diverges into two parallel 3-carbon pathways. Although two distinct molecules are formed, only one of them, G3P, can directly proceed to the next stage of glycolysis. The other molecule, DHAP, must first be converted into G3P before it can be further metabolized in the subsequent steps. An isomerization reaction quickly follows the cleavage step. The enzyme triose phosphate isomerase catalyzes the rapid and reversible conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. This reaction is crucial because it ensures that both 3-carbon units derived from the initial glucose molecule can be utilized in the energy-generating phase of glycolysis. At this point, the single molecule of glucose that entered the pathway has effectively become two molecules of G3P. Therefore, all subsequent reactions in the payoff phase occur twice for each molecule of glucose that is initially processed. This is where the pathway begins to yield a return on its initial energy investment. The first step of the payoff phase is a critical energy-conserving reaction. Glyceraldehyde 3-phosphate is oxidized and phosphorylated by the enzyme glyceraldehyde 3-phosphate dehydrogenase. This reaction forms 1,3-bisphosphoglycerate. In this step, a molecule of inorganic phosphate, or pi, is used, not ATP. The reaction also reduces a molecule of NAD+ to NADH. This is a significant event because NADH is a high-energy electron carrier. It can later be used in the electron transport chain to generate a substantial amount of ATP under aerobic conditions. 2 molecules of NADH are generated per glucose molecule. The newly formed one, 3-bisphosphoglycerate has a high-energy phosphate bond. This energy is now harvested to produce ATP. The enzyme phosphoglycerate kinase catalyzes the transfer of the high-energy phosphate group from 1,3-bisphosphoglycerate to a molecule of ADP. This process is called substrate-level phosphorylation. It results in the formation of 3-phosphoglycerate and the first molecule of ATP in the payoff phase. Since this reaction occurs for each of the 2,1,3-bisphosphoglycerate molecules, a total of 2 ATP molecules are generated. This generation of ATP exactly balances the 2 ATP molecules that were consumed during the preparatory phase. The rate of glycolysis is not constant. It is carefully regulated to meet the cell's metabolic needs. This regulation occurs primarily at 3 irreversible enzymatic steps. These steps are catalyzed by hexokinase, phosphofructokinase 1, or PFK1, and pyruvate kinase. These enzymes act as control points, allowing the pathway to respond to changes in the cell's energy state. High levels of ATP, a signal of high energy, inhibit the pathway. Conversely, high levels of AMP or ADP, signals of low energy, activate the pathway. This feedback system ensures that glucose is consumed to produce energy only when it is needed by the cell. Hexokinase, which catalyzes the first step, is the initial point of regulation. It is inhibited by its own product, glucose 6-phosphate. When the concentration of glucose 6-phosphate builds up, it signals that the cell does not need to break down more glucose for energy. This product inhibition prevents the cell from trapping more glucose than it can process through the glycolytic pathway. In the liver, a specific isozyme called glucokinase performs this role. Glucokinase has a lower affinity for glucose and is not inhibited by glucose 6-phosphate, allowing the liver to process large amounts of glucose after a carbohydrate-rich meal for storage as glycogen. The most important control point in glycolysis is the reaction catalyzed by phosphofructokinase 1 or PFK1. This enzyme is allosterically regulated by several molecules. High levels of ATP act as an allosteric inhibitor, binding to a regulatory site on the enzyme and decreasing its affinity for its substrate fructose 6-phosphate. Conversely, AMP and ADP act as allosteric activators, reversing the inhibitory effect of ATP. A potent activator of PFK1, especially in the liver, is fructose 2,6-bisphosphate. Its concentration reflects the hormonal signals that regulate blood glucose levels, integrating glycolysis with the overall metabolic state of the organism. The final regulatory enzyme is pyruvate kinase, which catalyzes the last step of glycolysis. This enzyme is also allosterically inhibited by high levels of ATP, which signals that the cell has sufficient energy. Alanine, an amino acid synthesized from pyruvate, also serves as an allosteric inhibitor, indicating that biosynthetic precursors are abundant. On the other hand, fructose 1,6-bisphosphate, the product of the PFK1 reaction, acts as a powerful allosteric activator. This is an example of feed-forward stimulation, an accumulation of fructose 1,6-bisphosphate signals that the pathway is active, thus preparing pyruvate kinase to process the incoming intermediates efficiently. The remaining steps of glycolysis focus on rearranging the molecule to set up the final energy-yielding reaction. The enzyme phosphoglycerate mutase catalyzes the relocation of the phosphate group in 3-phosphoglycerate. It moves the phosphate from the third carbon to the second carbon, forming 2-phosphoglycerate. This is an isomerization reaction that is readily reversible. The purpose of this intramolecular shift is to position the phosphate group in a way that allows for the creation of a high-energy phosphate bond in the next step. The enzyme requires a small amount of 2,3-bisphosphoglycerate to be present to initiate the catalytic cycle. Following the mutase reaction, the enzyme enolase acts on 2-phosphoglycerate. Enolase catalyzes a dehydration reaction, removing a molecule of water. This creates a double bond in the substrate, converting 2-phosphoglycerate into phosphenyl pyruvate, or PEP. PEP is an unstable, high-energy compound. The removal of water dramatically increases the energy potential of the phosphate bond. This sets the stage for the second substrate-level phosphorylation event of glycolysis. This step is crucial because it generates the high-energy intermediate necessary for the final synthesis of ATP in the pathway, moving the cell towards a net energy gain. The final reaction of glycolysis is catalyzed by the enzyme pyruvate kinase. This enzyme facilitates the transfer of the high-energy phosphate group from phosphenolpyruvate to a molecule of ADP. This reaction produces a molecule of ATP and the final product of glycolysis, pyruvate. This step is highly exergonic and essentially irreversible under cellular conditions. Since this reaction occurs for each of the two PEP molecules derived from one glucose molecule, it generates 2 molecules of ATP. This brings the total ATP produced in the payoff phase to 4, resulting in a net gain for the entire pathway. In summary, the overall net reaction for glycolysis is the conversion of 1 molecule of glucose into 2 molecules of pyruvate. For each glucose molecule, the pathway consumes 2 molecules of ATP in the preparatory phase. It then generates 4 molecules of ATP and 2 molecules of NADH in the payoff phase. Therefore, the net energy yield from glycolysis is 2 molecules of ATP and 2 molecules of NADH per molecule of glucose. The pyruvate and NADH products can be further metabolized in other pathways, such as the citric acid cycle and oxidative phosphorylation under aerobic conditions to generate a much larger amount of ATP.